Hello everyone, my name is Aleksandr. I will today present you a small presentation, small talk about the artificial intelligence, Zarr and special AnnData format for digital pathology in general, but of course this is also applicable to cancer. Few words about me. By my bachelor's I am a molecular geneticist, but starting the bachelor's I already became the quant at hedge fund. Later, I applied to the computational biology, and currently I'm an MSc in computational biology studies. So my main work at BostonGene is basically the team leader of the machine learning and artificial intelligence applications, particularly to imaging. We are working with the current state-of-the-art neural networks, AI, and a lot of other stuff to basically help to treat cancer. And let me start my presentation with, I guess, with what already everyone saw. So what I will be speaking about is basically on this image, because this is an AI hype. This is like ChatGPT. We will be sometime replaced, but if you work in this sphere, you got a lot of articles to read. You got a lot of information. Some of this is complete trash, and currently we are overwhelmed. So here at this presentation, I tried to include more of, I would say, common language, just to not overwhelm you. But of course, there would be a code and some technical details for those who are very interested in a particular sphere. And let me start with a few slides. So that's the statistics of the current leading share of death from major causes. This is statistics for 2021, and this is for United States. And here you can see that cancer is at the top 2 place, and most of these diseases, they basically share something in common. And since we're in Armenia, I of course included the Armenia plot. This is for 2022. The statistic is the same. Cancer is the second leading case. However, the cardiovascular disease also dominate. And what is so common between these diagnoses? This diagnosis is basically the fact that approximately 17% of all of these diagnoses, they are pathology-based. So what does it mean? Whenever you need to finally diagnose some person, some patient with particular treatment or assign the treatment, you need a pathological examination. Either you are taking the biopsy, in this case, for example, if lymph node was resected, you are taking part of it, putting it under the microscope, and then study it. Like what happened exactly, what is wrong here, what's wrong with the tissue. Same for example for cancer. For cancer, you cannot just simply remove some part of the tumor. For example, brain tumor are completely irremovable. I mean, it will damage the patient itself very hard. In this case, there is existing the core needle biopsy. Whenever you are putting the very, very narrow needle inside the tumor, taking the biopsy, taking it back, and then also still you're analyzing it under the microscope. And that's the cornerstone of the whole pathology, because that's basically what you obtain from the patient. Whenever you have the primary site, cancer, whatever lung diagnosis, like any pathology-related disease, you always finish with these what's so-called glasses with a biosample. And in this case, for example, we are taking it, and then you need to accurately examine all of these glasses to understand what are the case of the disease, what exactly driven it. And for example, for cancer it's wrong to tell that cancer is one diagnosis. It's basically a huge group of diagnoses. They are all very different, and you need to study these glasses pretty thoroughly in order to understand what exactly are type of the cancer, why it's elaborated, and how you need to treat it, because otherwise you will simply kill the patient. And only by doing this procedure, once again, for 17% of diagnosis, this is day-to-day routine, which is done in most of the hospitals, most of the clinics. That's what each pathologist are basically assigned with. This is just an examples, but this is one palette of these glasses which are ready for staining. I mean, each of these glasses is a biopsy, and each of these glasses, it should be examined. And usually there is like, if this cancer center or hospital is very big, you can have up to even 1,000 such samples, 1,000 of such glasses per day. So this is absolutely crazy number, because imagine the number of pathologists which are well trained you need to have to basically just to be able to assess them. Then you need to start to treat people, and this is also complicated because you need to purely understand what exactly happened at this level, at the level of tissue, and this is like the huge complication. And another word about the pathologists, these are very educated, very trained persons, but they learning through all of their lives. And to be able just to hospitals let you in and just to let you examine these glasses, sometimes you need to spend 10 to 15 years. 10 to 15 years, which is huge. So imagine that all of these people, they are well trained, they of course cost a lot, and you simply cannot somehow help them currently at this state. But something changes, and I will tell you what changed exactly. And this is already the portion of this tissue sample under the microscope. So that's the example of breast adenocarcinoma. If you are not well-trained pathologist, you will not understand anything. Just simply, this picture, this image will not tell anything to you. But here, for example, there is a lot of cancer, there is a lot of lymphocytes. So there is already a lot of information about how to treat this person properly. Because once again, all cancers, they require the particular targeted drug. And if you just assigning some wrong drug to person, it means that you launched a race who will survive, cancer or patient. And if you miss the drug, in most of the cases, patient would not be a winner here. So there is a lot of questions which our pathologists are usually asked for example, in order to help patient, while you examine this biopsy, you need to understand, first of all, is there any pathological tissue? Is there anything wrong at all? And sometimes this piece of the tissue which is wrong could be very, very small. And if you miss this, it means that you just, I would say kill the person, but that's completely not true. But still, you miss the diagnosis, it means that the treatment would be late. And this is an unfavorable scenario. Also, you need to understand what percentage of tissue are pathological, like how severe the cancer are, how severe like some of the lung diseases. And you just mark this area and understand how big it is. Grade stage, this is cancer related. You also need to understand, examine each nucleus of the tumor and then understand how it is formed, which shape it got, and how severe the cancer are. Any other clues? Because usually there is like additional information on the tissue, for example, presence of lymphocytes, the pattern of their distribution. Like there is a lot of additional scenarios, and you need to just simply find them. And believe me, this is very hard. But these samples, they're usually huge. Like in the context of these glasses, let's return to this slide. You can see that likely this is very small piece of tissue, which it looks like it's very simple to examine them. But that's actually not true, because actually samples, whenever they are scanned, like there is a 40 times magnification and there is a resolution at the level of each individual cell nucleus. And this step, it will look like this. So the pathologist would just examine all of this big piece of tissue, and in any part of it, there could be an issue. And if you missed it, you have a problem. Not you exactly, but patient got a problem. Usually it looks like this. So you got these microscopes. Currently they are already fancy. They are not a light microscope, but electronic ones, digital versions of it. So you just can have a pretty big screen. But later these days, it was common to use the light microscopy, which even was more exhausting. And that's, once again, at the level of each individual nucleus, you need to assess this tissue, which is pretty hard. And the problem even is much harder, because at the first glance, okay, let's introduce AI. Like, why the hell do people need to assess them manually? Let's just make some neural network, train this neural network, which will find this area for us. Looks like simple. This is like techno-optimism, but still there's the huge problems in this sphere, especially in the digital pathology. Because once again, this is just glasses from one patient, which was assessed from one patient, different localizations, different different tissues, but still each of them needs to be examined. If we would have some system which would be able to do something like this, for example, this is just an example how the neural network should perform. You have some tumor areas which are outlined. You have stromal area, lymphocytes. This is additional clues and additional information about how to treat people, and a little chunk of normal tissue. So you can see that some of this area are not covered, but even at this resolution, this is still fine. Like, if you will produce these results to pathologist, everything would be good, because you cannot just simply skip this piece of tissue. But that's, of course, not so easy to overcome this problem. Of course, if we would be able to, it means that AI brings a lot of benefits. First of all, because of instant data processing. I mean, you cannot put one pathologist and assign 10 glasses in parallel to him. This is simply impossible. He can analyze only one glass. AI is not like pathologist. He can be assigned to hundreds of samples, and still it will successfully process it. This is like common situation, but pathologists, I mean, need 10 minutes for examining the glass. AI needs approximately 20 to 30 minutes. The time difference looks like pretty big, but still remember the parallelization. AI is highly parallelizable. In this case, you can just assign a lot of glasses here. It is about the increased accuracy, because of course, whenever a pathologist looking through these glasses, through this tissue for several hours, he's definitely exhausted, he or she. He could miss something just because of this tiredness, because of the red eye, and that's also a huge problem, because you cannot just burn out your staff and assign pathologists to work, I don't know, 12 hours a week, 12 hours a day, which is nearly impossible. And of course, if this would be integrated just in clinical practice, just imagine the biopsy was taken, the glass was prepared, stained, and then you got a report from AI. Looks like pretty simple, but there is tons of the problem. Here it is an example of the previous slide, once again, and here, for example, there is a result of annotation from 4 different pathologists. And each of them was asked to manually segment or label the part with the normal tissue. And here at this very step, we have our first problem. Because pathologists, they're humans, they got their own perception. They was trained for 15 years, for 20 years. They all got their own perception. And for example, here it is nearly impossible to say which segmentation is correct and which is incorrect, because all of them, they captures all of the normal tissue. But in this case, for example, for training the neural network, this is pretty critical, because if you will have part of the data set labeled by first pathologist, part of the data set labeled by the second pathologist, it will simply fail to recognize the normal tissue in this case, because all of these predictions, they are very different. And this is the major issue, because in average, this is the results of the overview. On the x-axis, there is a ACC, ICC, or kappa value. This is the agreement level. 1.0 means the perfect agreement. 0.0 means absolutely no agreement here. And what's between the 0.4 and 0.6, it is what's so-called moderate agreement. It means that, okay, some of you agree, some of you are not, but still the results are pretty different. And here, what is interesting, this is the results, this is different data sets, manually labeled by pathologists, and they was asked to perform some kind of a task. And then we are measuring what are the average concordance rate, or what are the average agreement between pathologists, and we obtain the number around 55%. This is like the mean in the industry. And it means that we completely cannot use simply any kind of such annotation, because in this case, we will train our neural network to be a pathologist, one particular pathologist. I mean, if you have several of them, you will became the median pathology, all out of them, and that's it. And that's a huge problem, because you cannot train these algorithms based on such labeling system. And usually, most of the studies, they are failed, and they are failed in transfer between hospitals. For example, if this algorithm would be trained in one hospital, it means whenever you're applying it to the hospital number 2, it stopped working at all. And no one knows what's the problem, but this one of the problem, because in hospital number 1, pathologist and pathologist from hospital number 2, they are in disagreement. Like they're not agreed in what are the proper label, for example, for this image or for this area. And in this case, this is the pretty awful graph as for me, just because here is the whole publications mentioning AI and the publications with AI in biomedicine or medical imaging. And here we got a huge problem. As you can see, there is a small race at approximately 2015, and I will say why it happened so. But in general, you can see that the trend is pretty poor. The whole applications of AI in biomedicine is completely wrong. Like no one widely apply these algorithms to the biomedicine to obtain the pretty good results. And by the way, the COVID is also demonstrated exactly the same situation. Like during the COVID, of course, a lot of neural networks was trained to recognize the COVID based on the rentgenology, based on CT, a lot of other stuff. And it was proven that out of 3,000 models, only 2 was trained correctly, like 2 out of 3,000. All of others was overfitted, was incorrectly trained, was incorrectly labeled, and that was the major issue. Like we here now at the year 2024, and even for COVID, we are unable to adequately react with this models. What happened in the 2015 actually? This was the first glow of the artificial intelligence in biomedicine, because before of the 2015, it was pretty widely stated that AI will never cover the biomedicine. It cannot do anything with pathology. It cannot work with anything medicine related. And we need just train our pathologists and rentgenologists more. That was the base level. At 2015, there was an UNet architecture emergence. Maybe some of you know what is this. It is like the U-letter architecture of neural network, which first allowed this kind of the segmentation. So at this very level, this was the first neural network which was able to tackle this problem, like only 2015. And in general, the trend is following that in biomedicine, we are late in maybe sometimes 3, even 5 years. I mean, if something would be brought by data scientists in our world, I mean, some new architecture, some new approach, in biomedicine, it would be taken only after 3 to 5 years. And this is the huge also a huge problem because, of course, data scientists, they are not so interested in pathology. I already said that we lack the good annotation, we lack the good labeling, we got a lot of hospitals, and additionally, the problem is the very basic problem. The slides which we are analyzing with this tissue, they are huge. They are not like huge, they are enormous in their size. And just imagine this one particular image, this is, I would say, the mean size of the slide. 16,000 pixels to 40,000 pixels and 3 channels also. So in this case, we are simply unable to somehow process it by neural network. We cannot put the whole slide in neural network and ask, hey, give us please a segmentation. This is impossible due to size. And that's also not a problem, but the problem is pathologists got their day-to-day basis routine. That's the statement number 2. The statement number 1. Number 2 is that they do not want to be replaced by some kind of the artificial intelligence, and they simply are not very grateful whenever you're asking them to provide the labeling for you. And of course, imagine that if you have such big slides, just to manually label some of the particular regions of interest, it could take up to some days. And once again, this is artificial intelligence. We need thousands of such slides. So all of these pathologists who are board certified, who spent 20 years of their life just learning, they come to the point that, first of all, this is very expensive. Secondly, this will take just huge amount of time. You just cannot process this in some short period of times. And thirdly, they are in disagreement. But that's not all. As I already said, sometimes the area, target area on the slide it could be approximately 1% of or even less. For example, this slide, this is 10 millimeters, a ruler, and whatever marked with the yellow here, this is the target area. This where the pathologist needs to come and just manually label this by their hands. And imagine just trying to find all of these regions based on this one particular slide, and there's like 100 slides more. Of course, a lot of pathologists just frustrated, and to produce such kind of annotation, you need to be like highly trained. This is only available for pathologists who spend 30 years of their life just analyzing these slides. And one particular issue, it is a technical one, because maybe you already heard about the what's so-called batch effect, and batch effect in the artificial intelligence is pretty severe, but I mean you can still somehow tackle it. You got a lot of ways how to work with it, and in general it's completely fine. But here, this is the same piece of tissue, but this is scanned with the Philips scanner, and this is scanned with the Apereo scanner. And as you can see, there is a huge difference between them just by staining. Like this is pretty pinky slide, this is slide which is already something like purple. And this is diagram with the hue and saturation value. And if you will check the Philips and Apereo, you will see that this distribution, these kernel density plots, they just showing you that they are pretty different. And maybe for human side, this is not a problem, just completely not a problem. But for AI, we need to normalize our inputs. And whenever you will use the normalization values, I mean mean and standard deviations for channels, for Philips, based on Apereo scanner, it will not work. You like shifting the distribution, and at this point, you're just unable to produce any relevant outputs and any relevant conclusions based on these slides here. So we here in the digital pathology area, we are in a huge trouble basically, because first of all, it is very hard to obtain the high quality annotation, yes, because pathologists are resistant, they do not want to be replaced, they are in disagreement, slides are huge, that's the problem number one and two. Problem number three, that these slides, they actually normals, and you still need somehow to just process them. And obviously we need a mechanism. Of course we can do it by crops and with cropping, but then there is a good question, because tissue, this is, I would say this is an interconnected material, and whenever you are just cropping it, you just losing some of the context. And this context, it could be very important. Like if you lack this context, you can misclassify the cancer with some normal tissue. And I mean, that's without context, that's completely fine. And finally, it is also very computationally expensive to process such files and process such data. Just because of this, once again, these slides are huge, and you cannot just simply put them into the your personal computer, and this will not work like this. So what we need to do, and what are the problems, is that first of all, we need a lot of data, and currently more than 90% of such digital pathology data are unlabeled completely. Like it's okay if you have some top-level organizational diagnosis, like this is a breast cancer. That would be fine, but usually this is just a glass, a piece of tissue, and nothing at all. Like what to do here, how to train an algorithm. And for example, biggest ImageNet data set, which are commonly used in computer vision, it consists out of 14.2 million of slides, of images, with size approximately 3 to 224 square side. This is channels, width and height of the images. And this data set have nothing common in pathology. Like you cannot somehow transfer this knowledge, transfer this data set, and algorithm trains on this to these slides, to this pathology. This is also impossible. So here we was in trouble, and I would say that until year 2020, in digital pathology, there was a complete stagnation because of all of the problems I mentioned. But what actually happened? The general idea stays the same. Like we have our image, then we have our encoder, Encoder could be a function, encoder could be a neural network, which is also a function basically. And this representation, which you obtained based on this encoder, they could be used for the all downstream tasks available. And this is the definition of the foundation model. Like foundational model, whenever you're applying the same modality of data, they output you the consistent output, which you can use in your downstream tasks. In this task, for example, here we got the kitten image. Here there is a depth estimation. Here there is a semantic segmentation. And here there is a style transfer, for example, whenever we are trying to train some of the stable diffusion and generative AI. What we also know is that during the late 2010s, there was an emergence of the large language models. And large language models, they are also most of the time, they are trained in what's so-called the self-supervised learning procedure. So what is this basically? You have the raw text, then you pass it through the model, which is trained during the self-supervised learning training regimen. And then you are going to a supervised learning and fine-tune it to some of the downstream tasks. So what are the examples? Examples are simple. You have the text corpus. For example, nothing is impossible, even the word impossible saying I'm possible. And then what you need to do, what is our self-supervised learning? It's like whenever this neural network trains itself. Like you do not need to control it, you do not need to input something else than the raw data. And then it just learned that after word nothing, it is very likely to obtain word is. Then, if you got the combination nothing is, it is very likely to obtain that next word is impossible. And then, based on these corpuses and based on the large language models, you are just training these models. You are pre-training them basically with the self-supervision. And this is also a pretty good example of how we can train such models. That's randomly masking approach, it's also a self-supervision. For example, if the initial word sentence was the quick brown fox jumps through over the lazy dog, you can just randomly mask some of the tokens, some of the words, and ask the neural network, please predict what is the most probable word here. And then you can have it training, and the neural network will at least learn some baseline representations. Then you are going to continue with the supervised learning and fine-tune it up to your task. And now we have such kind of the ChatGPT, Sonnet, Claude, and a lot of others. But the question is how to apply it to the images, because images, obviously, it's not a text. And you cannot just simply ask, what is the next pixel? It's basically a nonsense asking the neural network what is the next pixel, because it depends on the context and it depends on what are on the image. And in this case, at year 2020, basically self-supervised imaging, learning and imaging emerged. It started with the paper which was called Biol, Bootstrap Your Own Latent, and it included the following procedure. Like here now, we are trying to repeat the logic which comes from the large language models. Like we are taking the image here and then taking two random crops out of it. Like at the random positions with different scales, but still the idea is to take two different crops from the same ideal image. And then what you are doing, you are having the what's so-called target network and online network. It then evolved to the student network and teacher network. And you are just passing, for example, this ResNet. It's the state-of-the-art computer vision model for the image classification, segmentation, like whatsoever. You are taking this neural network, passes two of this representation here through all of them with a little modifications, and then just asking that this representation and this representation, let me switch this slide, representation 1 from first neural network and representation 2 from the second, they should be similar. And the last function here, it is the similarity of two representations. Take the cosine similarity, like whatever distances you want, but still just minimize this dissimilarity. And this neural network would train. And here it is just details. This is L2 norm, which just minimizes the magnitude impact. In this case, neural network just cannot apply magnitude, it cannot just vary the absolute numbers of vectors. And we are making this absolutely incapable to this. But in this case, our neural network starts to finally train itself. Because what we are actually asking for, like let's go to the next slide, to this slide. This is the example augmentations for both of these crops which could be applied, like crop number 2, crop number 1, and we are applying all of these crops to these images. And here, as you can see, these augmentations called global because they are very severe. Like some of the, for example, Sobel filtering, leaving you only with ages, and you lost all of the context of your image. But still, if you are as human can see that here still is a dog, and I can see it clearly here, it means that neural network also should see that here there is a dog. And I mean, train yourself to recognize that there is a dog here. And it happens that, why it is even training? This is because of the very crazy reason, because just to remove this, increase this similarity between two representations, the only way is to discard all of this representation and start learning something meaningful about this object, which is on this particular image. Just imagine that each of this crop could be crop number 1, crop number 2, and you are just passing them through the network. And that works based on this algorithm. Neural network started training itself in the computer vision, which was nonsense until the year 2020, because no one even knew about this approach, and this technology is only 4 years old. I mean, this is very recent technology. And this is also very interesting because the approach I showed you, this is called BIOL, and this is red lines here, and supervised network with the same number of parameters is in blue here. And as you can see, neural networks which are trained with this approach, with self-supervised learning, they are even capable to overcome such models which was trained in a supervised manner, which is also crazy because like how this could happen whenever you are just randomly passing crops of the same image. And I mean, you of course should have a lot of images, but still, how it is even possible? But the training here, and this what basically first of all changed the whole area of computer vision, because now how you usually apply this, if you like have your very unique and I don't know emergent data set which only you possess, in this case you cannot just take the ResNet by default with pre-trained weights, and you cannot put it through your samples and obtain something meaningful. But here, if you got a lot of pictures, just train it in a self-supervised manner. You also can take pre-trained networks here and just start passing your data here. And it will learn and it will train and even be pretty close to this what's so-called supervised learning whenever you are passing these labels. And that's, once again, that's also a complicated procedure because not everything is so clean about it, and not everything is so easy. First of all, there is a problem about the batch size. Using this approach, you cannot have the small batch sizes, and in this case, you should have enormous batch size. So imagine you should pass into one single forward propagation step, you should pass ideally 4,000 samples. And this would require you sufficient GPU resources and GPU memory, because otherwise it will just drop in performance. And this is clearly seen, and this process is very rapid. Also, the augmentations or transformations which you are applying here, this is particularly one, this is this set of what you can apply here. The whole training procedure is heavily relies on them. I mean, if you will remove the grayscale, it will drop in like 5% of the quality. If you will remove the color augmentation, it will drop in another 5%. So it means that for each of the data set, it is critical to fine-tune and find this proper set of the augmentations, because otherwise you will have situation like this, and this system will not train at all. And also the bad news, you will not even observe it because the loss will converge, but still there would be no meaningful and representative embeddings coming of these neural networks here. And of course, like in year 2017, we had a new article image worth of, I don't remember exactly, 16 to 16 patches, if I'm not mistaken, about the visual transformers. So of course, the large language models, the initial transformers here, they arouse the visual transformers in the computer vision sphere. Basically, this is a BERT architecture for the large language model modeling, which was just tuned based on pictures. So here the idea is you're just splitting these patches, your image in patches, and just fitting them into this transformer with the attention layers. And it will work. And why it's important? Because attention layers, they actually can tell you something about the regions and some properties of the objects on each of these pictures. For example, here you can clearly see the bird. Here you can clearly see a brush, a sail, bicycle, and here all other pixels which are not so meaningful. Of course, you can just discover into this attention. And basically, current research in computer vision, this is based on the transformers architecture. There is a lot of applications of this. For example, from the business side, you can apply this well-trained visual transformer to your website and then just see, like, from the human perspective, what is important and what is not. And that's how also the computer vision works. And here, it was a question how fast we will adapt these self-supervised learning methods to these transformers. And the answer is that in year 2023, so just imagine this is 1 year old, we got the what's so-called DINA V2 method, which is basically suitable for training these state-of-the-art visual transformers. This got a little modifications, like still we have this image initially, X is our image. We have two crops, X1, X2, with different set of augmentations. Then we just renamed this network to student and teacher. And then we also additionally just applying some of the techniques such as centering, softmaxing, and now we are just optimizing the probabilities or the cross entropy. And this, just for those who are interested, student parameters optimized in such a way that only global augmentations go through the teacher network. All augmentations goes, of course, through all of the networks, and then you are just trying to calculate this cross entropy. So what do you want to obtain exactly is that for student and for teacher, these meaningful pixels, this meaningful information should be the same. And this is just the DINA version 2 clue, which is widely used. And now I would say that this is the state-of-the-art method in self-supervision in the computer vision. So also, ResNet architecture could be trained via this approach. It means that if you do not have enough GPU memory, you can just switch to the ResNet and basically nothing will change. You will still use the same architectures, but only change the neural network architecture itself. So now, the only one question which is left is how to basically apply this self-supervision approach to these images, because they still huge, and we need to somehow manage somehow to process it, because still we need an answer. Where exactly the tumor located? Where exactly the lymphocytes located? And a lot of such information. And what was proposed is basically a pretty interesting idea, but what we need to do, we need to stop treat the image as the whole part of it. Let's just split and rechunk it into the pretty small grid, and extract all of these crops with this kind of tissue, because at some level of magnification, at some resolution, with some side of the crop size, you will have literally only one type of the tissue on this slide. And this will work up to some point, but it happens that this point is pretty enough, like you can have the higher than 95% of accuracy. Then what you also can do, this is pretty interesting approach with the masked global crops. Whenever you're splitting this crop into even more crops and just randomly masking it out. And you are asking the neural network, please just recreate what it should be. What we could observe here if there would be no masking at all. So once again, here we just came with the same idea with the large language models. So this one algorithm Imagine that this is replaced completely the next best token prediction, this one, and this one masking just repeated what we observed previously. And this masking technique, by the way, was taken from the Visual Transformer article, which is saying that, okay, you could mask up to 17 to 80% of the whole image, and you still will obtain the meaningful representations. I mean, if you got the original, then you mask some part of this image, and this is the result of the Visual Transformer work. It means that it is able to recreate what, like, in low resolution, but still it got the representation and got the idea what should it be and how it should look like. And this is my favorite part, because here, for example, with 85% masking, here you completely hidden this tomato out of the initial image, and there is no this tomato. It means that if this would be overfitted, it would recreate this tomato, but this is not recreated this. It means that this is pretty well trained, and yes, we finally got something meaningful. This is also with the shape of this vase. If you will notice that these shapes are pretty different, because we masked a lot of the very big parts of this vase, and it just recreated how the average vase would look like. And this approach works. So then what we can do, we can train our network in this approach. So we are just tiling our image into very small crops, passing it through the network, training it with the self-supervision. And then what we can do, let's just simply put the representation on the dimensionality reduction methods. PCA, UMAP, use whatever you want. And it happened that all of these types of tissue, they are pretty well converged. So for example, here, there is adipocytes, which is fed. Here it is the background artifacts. Here it is some kind of debris, lymphocytes. So here you can better see it, because there's different types of tissue. They are grouped up in some kind of clusters. Here it is much bigger picture here. So here is lymphocytes. Here is some kind of a tumor. Here is stromal tissue. Here is muscles. So all of this tissue, they are just diverged. And now what you can do, let's simply perform a clusterization procedures. So let's say that globally now, when you need your pathology team, and the only point when you need it now is just to assess the quality of this final image, like how good your neural network in just linearly delineating these different types of tissue into different clusters or different parts of it. And here it has worked pretty good. And finally, how now the result could look like. And the main research in the digital pathology, they are now here in exactly this analysis. So here we have the piece of the tissue itself. This is huge one, once again 50,000 pixels to 50,000 pixels. So this is the huge image. And this is, each of this square is just the crop we took initially, went it through the network, and obtained the presentations. And finally, just use the clusters nomenclature. You can even visualize some part of this tissue, like what are the most common representatives of this. And now you can have just the answer about what types of the tissue, how they arrange, how they interact with each other, and even find some of the very small different types of tissue. Here they are in orange, which are pretty distant. And this, for example, could be a tumor, which is then is non-observable by human even. And finally, this is also a pretty good picture, pretty beautiful picture. This is how it could look like, and this is how it looks in, for example, our analysis. This is also, once again, this is the huge piece of tissue, which would require you a sufficient efforts and a lot of basically manual labor to annotate this. But here, yes, because this neural network is pretty consistent, this is just a huge zones with some kind of a tissue. Here you can see some kind of this, and this structures, which is also visible even in this picture here. Additionally, for example, in this zone, I don't know whether you see it or not, but there's two parts in this cluster analysis, because of course there's like necrosis zone and lymphocytes infiltration, and we can clearly see that there is something different. There's different types of tissue. And now, what we need to do simply, we need to ask our pathologist to assess, to just basically assign each color of these clusters. I would say it's in simple terms, which colors means which type of tissue. And at this level, we will able to output the whole decomposition of a tissue. And the biggest problem now here is only about the training of such neural networks. Because this is 3 different data sets on the x-axis, and on the y-axis there is a metric, accuracy or ROC-AUC. And here you can see that the coolest part is that you can fine-tune such architectures, once again, with this self-supervision. It means that you can train the pre-trained network on the pathology data set, take your data set and fine-tune it in only 74 GPU hours. And initial networks was trained at like 12,000 of GPU hours, which is crazy and will cost you tens of thousands of dollars. So even now in this aspect, but still, of course, you still need a huge batch size, which will require you to obtain a pretty big resources cage. But overall and in general, you're obtaining a pretty good accuracy, which was otherwise non-available. And even problem with scanners, whenever you have different hue and saturations, is already passed because, okay, if you got this neural network trained on one scanner, okay, let's just fine-tune it on your scanner of interest, and it will work as a charm. So that's generally how now it works. And once again, this is how the current analysis looks like. And this is the main field of the research. This is the main point of the research, how the digital pathology would evolve. And looks like we will additionally interpret these clusters. For example, there is already a lot of articles whenever they are connecting these types of clusters, these different tissues, with the gene expression levels, or with some mutations particularly. It means that at some point, some of the driver mutations could be assessed on only the computational pathology level. You do not even need the next generation sequencing. You do not even need the some chip sequencing to understand what are the driver mutations present here. Because I mean, they changes this tissue too significantly, and you can analyze it and get this information based on this level. So, what are the problems which are left? So now, we do not need the high quality annotation. We only need just a lot of data, which presumably we have a lot. We just collected, all centers collected it, and you can just train it, and you are pretty fine. Pathologists do not agree with each other. This is not also a problem anymore, because once again, in the training procedure, there is also only the data scientist. And data scientists, they are usually pretty connected in the mathematical terms, in the data scientist terms, in AI terms. So you do not need them to provide you the annotation. Like, just invite them to assess this quality, these pictures. And once again, pathologists trained to visualize. Pathologists trained on the visual data. Whenever you're showing them this picture, they're like very happy because this is visual, this is beautiful, and this is pretty understandable by them, which is also pretty cool. But still, we have a lot of problems. First of all, slides are enormous. That's still a problem. And the problem number 4 is that we still need a lot of GPU. We still need a lot of resources just to process such data. And now, I will stop on this image, I guess, because we will dive into the next topic with Zarr. I guess we can have the questional QA session for the first part, and then I will continue. I mean, if you have any about this part with the computational pathology.
What are ethical issues regarding using AI in treating cancer? Are there any such considerations that should be taken into account?
Yes, thank you for your question. basically there is a huge consideration, of course, because, for example, as developer of such systems, I do not want to go to jail. That's the first statement, I would say. Then it means that all of these systems, they are not standalone tools. These are the systems to help pathologists to annotate those slides. There is still a human in the loop. I mean, here, of course, we will show this image to pathologist, and it got a special viewers. It will just zoom in and assess that this is correct. And if pathologist would do not like some of the pieces, he or she will just re-annotate the slide by himself. But usually he just marks the I agree. And in this case, we're saying that according to pathologist review, because pathologist agreed, and in this case, pathologists are responsible. And he can manually control all of these steps, all of these visualizations, all of these results. And I mean, we're still keeping the human in the loop. And of course, in cancer, statement number 2, no one wants to go to jail. And in this case, all of these systems, they are aimed at just helping to get the right decision. And that's the whole story for now, because of course, in other way, it would be completely unethical to treat human with whatever AI output. There is a lot of additional considerations, like there is whole statement from FDA, how you should correctly apply it. There is GCP, GCLP statement, good clinical and laboratory practices you need to follow in order just to be able to apply such algorithms. It means that everything should be documented. You should know every potential pitfalls and issues. For example, if I don't know, if some kind of the hair puts down on this tissue piece, it could happen a lot. Just what would be the output? Will it say that all of this area from hair it is a cancer, and you will get the wrong diagnosis? So there is like huge topic, and I would say this would require a separate lecture to answer the whole topic.
Hi, I have a question. My name is Gev. It's not a question actually, it's just a fun fact. 3 years ago I was diagnosed with cancer, and a really difficult phase of my life started. And then in about a month I ended up in Germany, where I went I've gone through the operation. And then after that, my case was very rare case. And they were telling that this case, it was a colon cancer. And usually you have, according to the data, you have colon cancer when you are above 45 years old. Yeah. And I was 30 years old that time, 31. But I had a really severe stage of colon cancer. And then they said that you have to do a really in-depth genetic test. And I flew to San Francisco and stayed there for 8 months, and I went to Stanford Cancer Center, where I had to go through a really severe genetic test. The cost of that test was around $25,000, and what they do is they just take your saliva and your blood, and you have to wait like 3 to 4 months. But then they did some very funny American trick. What they did is like, you have to pay $25,000, but if you sign this form, if you give your consent, we can do this for free for you, but we will be able to use your data for our research purposes. And that test would cost you $150. So they went down from 24,000 somewhat to $150. And then I said, yes, of course, I want to do that. I can also call my relatives to sign under that. Yeah, give their consents too if you want. The reason I'm telling you is because the gentleman was asking about privacy and ethical side of things. And up until today, I'm receiving messages from Stanford Cancer Center where they're saying your data was used in this research number A, then this research number B. They don't declare in details, but you can basically request the result and how your data impacted that research study. And yeah, that's it.
Okay, thank you.
Just fun fact.
Looks
Thanks.
like you was a very unique case, and if you went through all of these procedures, it's like you got some case of, I don't know, very rare mutations.
Yeah.
And this is like, you need to ask the authorship in these papers, in this research, by the way.
Yeah,
It should
so
be
the
published.
funniest thing was that we did the same genetic test in Armenia, but then not the Germans nor the Americans accepted that because there is these BRAF mutations. Have you heard about BRAF mutations?
Yes, yes.
So
of
the
course.
BRAF mutations were not included in the Armenian genetic test,
Oh,
and they
in
all
colon
declined.
cancer,
And they say you
no.
have to have BRAF mutations.
yes.
And when you open my genetic test results now, it's just a whole bunch of just numbers, nothing like to understand. It's like a huge JSON of whatever big request, response of a request that I personally cannot read, but it's a very interesting, it was a very interesting experience for me. I was enjoying that 1 year. Everyone were super like stressed, but I was like super happy that I was going through that, and eventually I got everything fixed. So I'm happy, and I don't have the problem anymore. So
That's the best part, yes. That's the best part here. I can just congratulate you with winning this race. This is very hard. And by the way, just behind you, there is my colleague, Sitz, who works with the mutation analysis. And she can tell you about how these mutations, like this is the crazy procedure whenever you need to assess all of these mutations, just understand the pathogenicity of it, assess it from the ethics point of view. So this is pretty hard work.
Yeah, one last thing I want to say is that I have to speak in this? Okay. One more thing I want to say is that I don't want my all of the people here, our colleagues, to be like disappointed, but basically no one accepted the Armenian genetic test. And then that's okay, that's kind of okay. They were saying, okay, we kind of accept this by 80%. You don't have BRAF mutations, whatever ABC thinks. But the bad thing was that at the end of the genetic test from Stanford Cancer Center, they came back and to me and told me like, you are genetically cool, you don't have any problem, your kids are okay, nothing with it. The problem is, it's very sad, but that's the truth. The problem is the water, the air pollution, and the quality of your like ecology in Armenia. So go and figure it out in your country.
Okay.
And they don't recommend me to live in this country, but as you see, I'm
But you
back
end up
and I'm
here.
here.
This means yes, it's based on signature then, yes, then. This is, yes, whenever you do not have any mutations but still got cancer, this is also a pretty interesting research case because, yes, this is pretty complicated. Maybe, I don't know, maybe there could be technical issues, but generally, yeah, there is some articles which saying that, and this is like very rare condition, like extremely rare.
Yeah, I know, it's very rare.
This is like
Huge data of my...
they trying to find something, yeah, they try to discover it, yes. We're basically working with the same, so BostonGene providing the, how it called, CGP, comprehensive genomic profiling, whenever you got all of your genes, also mutations calling, RNA sequencing, like a lot of data, so also to help to find the treatment. And if you say you have the severe case, yes, usually this is the 3rd to 4th stages, whenever this is already complicated a lot, I would say.
Yeah, and what they were saying, one last thing, what they were saying is that, what everyone was telling is that basically because there is, according to the data that they have, there is no such an issue at that age. And if I have it genetically, then it means I have a Lynch syndrome.
Oh, okay.
So that was a huge thing. But then eventually they declined it.
Yes,
Yeah.
because they didn't
Yeah.
found the mutation. Okay, guys. So, if no more questions, let's go to the second part. So we still have two problems. Slides are enormous, and it is very hard to computationally process this data. And in this case, I will refer to these slides, basically 2 slides with the space. So whenever you're looking at this, I think usually you are easy terrified or mesmerized, depending on what kind of people you are. But still, we are interested not in the beauty aspect of this image, but we're interested from the point of the research, because NASA is processing these images like a lot. And usually on the daily basis, they are working with this. And for NASA, whenever you got these scans, whenever you got these images from the high technology telescopes, what you need to do and what you need to basically research in these studies, you are not interested in the whole picture at all. Like, this is not the point of the analysis. What you need to do, you need to zoom out, for example, zoom in this one particular star and check what are happening around this. Like, zoom in this cell, star, zoom in this star. And usually, we're just interested in zooming in and out and just analyzing and scoping the data. And now, that's basically the analogy. That's basically how also the tissue works, because how the daily routine of the pathologist looks like. You got the piece of the slide, and for example, now you can spot that here there is something strange. So I need to investigate what happens here a lot. And this is like 10x magnification. To get here, you need 40x magnification. So in this case, there is a software and special formats whenever you can do something like this. You are taking this region and just zooming in. And here still at some level of, for example, 20x, you don't not have any clue what happens here exactly, because you need to see the nucleus. You need to see how it's dispersed, how it's shaped. And this is pretty complicated, and then you will just go and zoom in even more. And finally, you will observe this image. And notice what's happened while we was zooming in and zooming out. So the quality of the initial image, I mean, you still here can see the details. Still here, everywhere you can see the details. You can see the exactly, I mean, at least some of the parts. They are not so blurry. And here especially, whenever you're going to the most deep level, this is still pretty good image. This has got very good quality. So basically, the images, they are not the image itself. Like, this is not a PNG. This is not a JPEG format. Images is basically, it is called the pyramid format, and it means that each level of this image is basically just the stack of the tiles. And they placed at the good position, and for example, this at the highest magnification, you simply got a lot of details, you simply got a lot of tiles. Then you are just using the downsampling, use interpolation, whatever you want, whatever is best suited for you. And you will have this stack of tiles for this image. Why it's important? Because here, for example, on the level 2x with 25 magnification, you cannot see anything. You just see the general patterns, but to assess what exactly happens with these cancerous cells and how they are shaped, what grade it is, what stage it is, you need to zoom out up to 400x of this zoom level, and this is pretty crazy, because this process should be very rapid. Like, you should immediately zoom in and zoom out, and it should load like in a second. And it means that what ideally do we need from this format for slides? And by the way, that's how the NASA formats looks like. They are also the pyramids. And NASA was the first one who adopted the Zarr format, which I will speak about a little bit later. So what do we really need from these formats to store these slides? First of all, this is the fast random access. Why? Because we need to go to some particular region very fast. Like in our computational algorithms, for example, if we observe tumor, we can have the image of the tumor, this region, particular region, and get it on the top of the mind of pathologist. It also should have a high compression, because this is pretty difficult to store all this pile of tiles, and the hard drives, they are pretty expensive. To store them all on the AWS, this is also pretty expensive. So the more compressed your data, the better there is. And ideally, and finally, you should lazily read it directly from cloud. I mean, if you got your clusters somewhere else, this image should be available for you just in the seconds. Just you do not need to load it into the memory directly, the whole image. You just need to some kind of particular region. Then let's extract it, and finally we will obtain what we need. So the standard image for the image format for the images, for the digital pathology, this is called TIFF, and this is not very well suitable. Why? Because first of all, it was invented to store the tax images, fax images, and printed images for A3 format. It means that this is basically not an image, but this is the correct collection of strips. And here it is visualized that there is a lot of strips, you can shuffle them, but still this came with the strips. And why it is so? Because initially, whenever you plot them on the, like this one here, this board here, you need to print this part here, you need to unzip it, print, while all others could be zipped still. And this was good and this worked well, but somehow this migrated to the digital pathology. And now what does it mean? If, for example, I need the region with garlic, I don't know, imagine that this is tumor, I got this region with garlic. It means that I cannot just get the image, the particular image, in memory with this garlic. I need to load this strip, and that's all, to the strips with all this unnecessary information. And for what reason I would do this if, once again, these whole slide images, they are huge, 60,000 pixels wide and height. It's just crazy amount of information, and I will just overwhelm myself and I will overload my memory. So then, what we need and what we basically adopted, and this is now the most common standard in the industry, this is called Zarr. And Zarr, it is based on the HDF5 conceptual model. You can just organize these multiple arrays, they are called Zarr arrays, into this hierarchy of groups. So this is structure of hierarchy, this is structure of groups, and all of this chunk data, I already say chunk, but still, this data, they stored as a binary blob with a pretty good conversion, like with a very high conversion. It basically allows you to treat each of these chunks separately. Like to read the chunk number 2.0, you don't need to read all of these chunks. Just get this particular chunk and that's it. And also this is allows you to parallel write and read. For example, if I am reading from chunk 0.0, it means that I still can write to the chunk 1.0. And it will work as a charm. Like still it is highly parallelizable. And by default, it is chunked mechanism. It means that your whole image, it is built up into such a grids with these 3x3 predefined chunks, which are stored pretty compactly, and you can just assess each of them independently. You need to take any of other data, and first of all, this is loaded lazy, and you don't need to spend any additional memory just to load anything else. And what does it mean? That if, for example, now there is my tumor region, and I need to get only these values, only these pixels, it means that Zarr will initially load these chunks for me, then just cut it down and let me in, and just give me that data. I will not touch any other chunks. I will just take all of this, and that's why this format also was adopted by NASA, because NASA obviously have a lot of information, much more than digital pathology, but still, they can allocate to very small piece of chunks, and then just take the necessary information. And that format is currently widely used. For example, if in this region, in my neural network, I need to take only the marked region, the segmented regions, it means that I accurately can load exactly these chunks, this part of information, and just keep the whole other slide. And this will save me just tons of memory, because now I can efficiently iterate. I can in parallel write, read, and do a lot of stuff, and this just because this file format allows me to do it. And the best thing here is that this format, this Zarr, it could be actually stored as the key value store, and it could be in memory store. It could be written in your directory. It could be built up to the zip file. It could be even just be accessible directly from S3 without any additional libraries, without any additional packages. You're just taking Zarr.open, pass to the S3 folder, and you are getting this data. So that's it. And basically, Zarr, it is the open format library, which means that you can do whatever you want. If you have your aims, you can rewrite it like in any way you want and chunk it, store it in whatever suitable format you have. And that's, for example, how it works. So Zarr, it's usually the upper folder with the root file. Here you can just say that this is sample number something whatsoever, and then, for example, this is the whole plate with the cells we need to analyze. Okay. Then what we can have here, we can just make the hierarchy whenever we will build this system and just put down number of row, column, and well just on spot. It means that by just putting down these values, we'll get the exactly needed chunk of information. And then this also gives you the resolution levels. So it's initially built in such a way that you can organize them with hierarchy built on resolutions. So here, for example, the downsampled pass with not so much tiles would weight something approximately 0.3 gigabytes, and the full resolution contains 1 gigabyte, which means that you can zoom in and zoom out. And if you do not need resolution, just go to the upper level of this pyramid. If you need resolution, go to the lowest level of this pyramid, and you will still have the initial data with all the details. And this is like pretty cool, because in pathology, once again, you need all of this nucleus, you need all of this information, and this just allows you rapidly to obtain whatever you want, process it, and put it back. And this would be just on spot, just on S3, on your data directory, like whenever you have a space, like any place here. And let's switch to the final topic of this speech about the AnnData. It's the final composition of all of these formats. And by the way, it also could be stored in Zarr or HDF5, the most common formats. This is basically called the annotated data, and it's specifically designed to contain this matrix-like data. What does it mean? It means that if you have, for example, each individual cell or each individual tile, you can just put it like a matrix with all of the necessary parameters. And additionally, what you can have here, you can put there a lot of other information which will also be completely packed, because this is in Zarr format once again. So here, for example, X is the main data matrix. This is your pandas data frame with whatever data you have, your values basically. Then you got what's so-called ops. This is the observations. This is any metadata of your rows from your matrix X with even, for example, coordinates of this tile, coordinates of this chunk, coordinates of this cell. So there is like also an additional data frame. So this is a data frame with data frames, which basically gives you a lot of accessibility and on-spot working with the Zarr format. .var, this is called the var names basically. It's called the metadata about all of your columns. So if you have something additional to add about your X, or maybe some observation matrix, or even your variables names and even your variables metadata, it's still contained in one particular place. And observation mapping, this is basically the most coolest part about it, and what we usually use widely in our research, is that this ops mapping, observational mapping, contains these multidimensional representations of our observations. And this could be a PCA, this could be whatever embeddings you want, for example, from Dino method, for example, from UMAP plot. Like there is information which is stored here, and this is still in one storage. And finally, this is .ans. This is what's so-called unstructured annotations. Here you can store any additional information, like whatever not mapped to the variable metadata, observational metadata, then you are putting it here. For example, palettes, mappings, whatsoever, like dictionaries, just put it here and it would be available. And once again, this is pretty good zipped, this is pretty good stored, and this once again could be stored in Zarr or HDF5, which allows you to compress this data like a lot. And All other components are pretty specific for a single cell analysis, basically because this AnnData was designed for a single cell analysis in bioinformatics, but still you can adopt it to absolutely any format you need. And today I will also show in code how it could work. So here it is all from aside from the presentation. I guess we can once again have the questions, if any, and then we will go to the little code section. Let's switch to something beautiful.
Okay, so my question is the format adopted by hardware producers, Zarr format?
But by what?
Hardware producers, scanner producers. Is it a common format for clinic healthcare providers? Or
For what once again?
is the Zarr format common for healthcare providers? Do they provide you data
Healthcare, yeah. Yes, so currently there is a format called OME Zarr. OME, additionally you are adding this is open microscopy environment. And yes, this is, usually you can convert all of your TIFF files, special formats for the scanners, SVS and DPI and others, in this OME Zarr format. And it will contain all of these pyramids and will allow you to rapidly access all of this point in data. So yes, this is now pretty widely adopted.
And one more question. So the annotation procedure, the pathologist who is doing the annotation should also contribute in creating the meta information for the particular annotation in terms of blocks. So is this realistic in applying in healthcare institutions?
Yes, you just can supply. I mean, in Zarr, once again, because you can have this hierarchy, first layer you can have the raw data, and then you can have, for example, annotation number 1 from pathologist number 1, annotation from pathologist number 2, and then you can do something with this. I mean, train algorithm, validated. Usually this data now used for validation of the algorithms. It means, yes, that, for example, I cannot pass this step if pathologist labeled that this is a tumor region. It means that in my clusterization and at all of my files, this region should go as a cluster number, I don't know, number 5, which is tumor, and this should be one single cluster. In other ways, it means that if I erroneously call this cluster a normal cluster, it means that I will not pass the internal regulations, first of all. And internal regulations of the healthcare, they sometimes even more severe than the FDA and all others, because it is widely known that if you release something which is non-ethical, which is non-approved, which got no documentation, you will likely be sued, and this is a pretty bad scenario, of course.
Thank you very much for a very interesting talk and a very illuminating talk. I was wondering, so you were discussing a system which helps pathologists to make a diagnosis. And I was wondering whether there are studies that show that it's actually useful to pathologists. I mean, to compare pathologists who doesn't use such a system versus pathologists who use such a system, and maybe the second group notice more tumors or take like time to make a diagnosis, something like that. Are there such studies?
There is a lot of studies in rentgenology and CT tomography, and in MRI. The best impact was in MRI, because in these cases, this is the 3D volume. You need to inspect manually and find any anomalies, which could be a tumor, something else. And in this case, yes, MRI operators who use the artificial intelligence systems, they've won like 5 to 7%. It's like the baseline, like 85% of complete correct diagnosis, and they just could increase it up to 92. And for rentgenology, it's the arms race right now. I saw the studies in which was claimed that yes, but rentgenologists who was assisted with the AI, they are also could win up to 7% of these diagnoses. I mean, based on rentgenology, and on digital pathology, the things are very complicated, because like only this year, 2024, this was the widespread. Like this is the first year whenever we see these systems, and there is no this, I would say, the whole study which will say that let's compare this and that. There is no such studies yet. I hope there would be some, but based on the experience with CT, rentgenology, it's likely to increase the quality of the annotations by pathology.
Thanks again for the talk. My question is about the AnnData. So did I understand it correctly that it's a format to store your annotation with the data? The X there is your data, like the image itself, not the
No. This is the addition to the Zarr itself.
Okay.
So here in Zarr, for example, we have chunk, we have tile with ID and with, for example, X coordinate, Y coordinate, where it's placed, and some metadata. So here in AnnData, in this case, we are using this observation mapping to just tell everything about these tiles, where to find it on the image, what our image it came from, and a lot of other necessary information. Here and here, we can store, for example, in X, in this case, we can store the embeddings from this neural network. So tile gets for this self-supervised network itself, and then here we can store efficiently the embedding for each of these tile. And I mean, the median amount of tiles is approximately 16,000 of these tiles. It means that there is a lot of data. And here also, because we are interested in drawing this data, how it's organized, we usually want to see and get the UMAP and PCA embedding, and then we also store it here. It means that rapidly, whenever we got the information about slide, we can just get to AnnData, got all of this information, got embeddings, got number of clusters. For example, this tissue came from cluster from lymphocytes, and then just get the final report or final output. That's just to store all of this data. It's just like complementary store to the Zarr file. Yes, to let us to keep all of this necessary information to these tiles. Because in Zarr it's impossible to keep all of this information and track it. And this is just not the pandas data frame, because pandas data frame we will have separate for data frame or storage for embeddings, separate storage for UMAP. And here we can see keep everything at one place.
Thank you. And if I may ask another question as well. So you mentioned that the pathologists themselves, they don't agree between themselves, right?
Yes.
So 55%, that's probably about early stages that they don't agree, or the stage wouldn't
This is,
matter?
yes, early stages and additional parameters such used in research usually. Yes, in diagnosis they mostly agree, of course. But I mean, whenever you are going to this very first image, I mean, somewhere here, yes, that's the problem, because I cannot say which one is true. Like every one of them marked the correct zone. But for neural network, this is the pretty disturbing situation, because you need to train it.
So, if you had higher agreement, if you could feed trustworthy data to the network, how would you use this additional information in addition to large quantities of unlabeled data? And you have a small labeled data. Is there a way to plug it in?
There is usually, for small labeled data, what we usually do, we have it, of course, a lot in our company. We use it mainly for validation purposes, which means that whenever we split this initial slide into the multiple chunks of data, like here, for example, then we want to calculate. For example, if pathologist, let's just tell it in simple terms, if pathologist mark this zone as a tumor zone, then it means that everything what is covered with cluster, I don't know, this one, is it true that most of this cluster consists out of tiles which contains tumor? And for example, if we got the high matrix, for example, if this cluster contains more than 95% of tiles with tumor, it means that yes, this is the tumor tile, tumor cluster, and we can assess it by it. But usually, yes, this data is very sparse, and of course, the main segmentation part, the main segmentation annotation is about tumor. But we're also interested about lymphocytes, about stromal component, about fat, whatever is here, necrosis, fibrosis, like there is a lot of other data we need to care about, and there is no such data. So this is very, very bad situation, but that's how the digital pathology evolves.
Can you use Zarr arrays as an NumPy array in multiprocessing in distributed systems,
Yes.
not in one processor?
Yes, it was designed as this. I will show you in the coding section just about it. It's naturally integrated with Dask and with Dask parallelization, with Dask array. It's integrated with whatever you want, basically, because once again, these chunks, all of them are independent completely. So you can work with each of them, read from one, write to another, and this is completely fine. Like, this will not spoil your data or analysis. Okay, so what we have here today, I will demonstrate you how this Zarr works. Let's first answer the question, and first topic, which we will cover pretty fastly. Let me just run all of these cells, and then I will get back to it. This is necessary imports. Let's do not care about it. And the opus magnum, basically, of all of this speech is this self-supervised learning networks. This is loaded, designed by Meta Corporation, Dino version 2, and this is visual transformer with patches. Scale even more, okay. Like this one. Okay. And this, how you can load this model. So this model is simply, it is a visual transformer. You have the first convolution stem, and then, where it is this, QKV, it is default attention layer. So here, and finally, this attention layers leads us just to an embedding with the length of 394 with layer norm all the time. And what we can do with this, we can simply transfer it to our CUDA, so our GPU memory, put it into the evaluation mode, and then here is just simply a function to generate embedding with this network. Once again, put this network embedding to the our GPU. And by the way, here I'm also demonstrating that PyTorch evolved, and starting version 2 something, they introduced this torch inference mode decorator, which works better than torch no-grad handler. It means that usually now try to use this one because it excludes backward propagation from any of these points, and this is the coolest part of it. And here just simulate the image and get the embedding. Now the interesting part, so this network is pre-trained on the ImageNet, and what we can do. This is the standard pipeline, since we have not a lot of time. I will not go dive deep into these details, but we can preload the CIFAR 10 dataset, which contains the images from 10 classes, collect the embeddings, collect the labels, and then, once again, what the self-supervision implies is that you are not using this label anywhere to basically learn, teach your network, but you are using these embeddings, for example, for UMAP. And then let's just generate the 2D UMAP and see what actually happens, how our network, this is only based on representation, so there is no any learnable layer, I mean the fine-tuned head. There is only the embedding itself. And on this level, different colors and different labels, you can see that they are pretty good separated just by this linear probing, and especially in 3D space. You can see that this network, which we never observed, your data, and this is CIFAR 10 once again, they already produce this embedding, which is pretty capable to distinguish between all of these classes. There is also a plotly graph, and I need a mouse to use it, but still, it is just to visualize that in 3D space, it works as a chart. So that's how you can simply use this network, and fine-tuning is also pretty simple. You are just getting your network into the training mode and just fine tune it, for example. Adding head, of course, but still, that's how it works. That's what's about the self-supervision. And now let's get back to Zarr. I guess it would be easier for me to do something like this. OK. So for Zarr basically based on its API, it is pretty similar to the NumPy. Because, for example, Zarr zeros with the shape of this array and predefined chunks with dtype will produce the array itself. The array itself is not accessible like if you will just follow the Z, which you are created just now. It will not give you any information about the data. What you need to do, get to d.info, and then you will have a lot of technical information about how this array is organized. In this case, this is shaped 10,000 to 10,000, data type integer 32, and by default, it already contains the compressor. It means your data is compressed on this very level, and as you can see, the number of bytes in this data is approximately 380 megabytes, but the number of bytes stored is only 345. It means that the storage ratio is crazy, but OK, this is a zeros array. We all know that zeros compressed pretty good. What's about the other formats? About the other formats is that, for example, you can put this data just on your local folder. You are putting zarr.open, putting the path to the array, mode write, once again shape, and chunks. And voila, you have your data stored exactly where you need it. So we wrote this into the example jar, then we read it, and by accessing this data, by this default notation with all of the Python objects, we got access to our data. We can reproduce it like this. So this, once again, this is a zeros array, and you can clearly see that, yes, that's it. Also, you can change the level of the compression. In this case, for example, I want BLOSC compressor with compression level 3, with special shuffle mode. And in this case, just create the Zarr array with defined compressor, and you will have it. Zarr array.compressor method gives you the compressor name itself. And of course, it is visible into the z.info method of the Zarr. And here it is our compressor. Then, about the chunks and about the parallelization itself. So you are opening this Zarr, pretty complicated 3D shape with the 3D chunks. And also it will work and we will populate it with some kind of the random data. In this case, it will take a time to create our data. I don't remember how fast it will take, how long. OK, it's taken not so long. So we got our data. What we can do, the parallelization, for example, could be achieved with Dask array. You are using Dask array, reading from Zarr one particular array, and they calling the Dask array mean compute, and in parallel you got your results. For example, here in this case, this is 0.5. What else you can do? For example, let's specifically update some part of this chunk, some part of this Zarr array. And by doing this command, once again, just on the spot, you got your result. In this case, let's check that data changed. And indeed, previous result was something like this. Now we obtain the different results. So because the data is pretty large, still we got a pretty low difference. And one of the final functions, it is the cool thing about the map blocks, which allowing you to apply your particular function to these predefined chunks. In this case, you are opening your array, you are chunking it, populating it with data, and for example, let's check what will change. Present min, then just normalizing each block particularly, and your Dask array map blocks will basically apply this function you defined here, for example, normalized block, to all of these chunks. Once again, to chunks, not the whole array. And finally, whenever you compute it, you had the exactly mean which you're predefined. And then let's check what happened after the normalization. And of course, before the normalization and after the normalization, we got nearly 0, so our normalization worked. It means that, yes, data in our chunks, this is pretty much changed. And also here we will have some magic in this function. But still, this is, for example, how the stores with images are organized. This is OME Zarr. This is what was asked about the healthcare industry. This is the standard for collecting this image data into these Zarr stores. So we have here two groups, 0 and OME. OME, it is metadata initially, and in 0 level, we have our arrays. And how does it look? 0, 1, 2, 3, 4, 5. This is the parameter levels, the initial parameterization application. Then, for example, we can get into the 0 level and see that, yes, this is pretty big image with the pretty big chunks, which are compressed pretty highly. And for example, here, number of initial bytes and number of bytes stored is with the compression rate, storage rate with 2.1, which means that we have saved just two times of our disk space by using this BLOS compressor. Same here. This is the first level of pyramid. The first level of pyramid is 2 times smaller than the 0 ones. And here, once again, the compression rate is approximately the same. And then, random access, we can get our data from this image. So just on the spot, I get some information from several chunks, and this gets to me just on the spot. Let's visualize this, how it looks like. And that's, for example, how my image looks like, which are stored in this Zarr array. Let's give the matplotlib time to plot, basically. Let's just, yes. And here we got the information. So that's the data stored in array. I just got it on the random access, just on the spot, and it's available to me. We can also go to the first level of pyramid, and it also will take some time to be drawn. And let's also check how it looks like, and that's also another piece of tissue. So just on the spot, based on this array, I just got all of this information. What's also good in Zarr, it is this nested organization. So for example, I can create my root and put down the store location. I now got my root, but there is no data here. So let's create groups. In this case, we implemented these hierarchy mechanisms. And for example, foo, it's equal root.create group with name foo, and bar, it's foo.create group with name bar. And in this case, I would have exactly my 2 arrays, which is kept in here. And let's populate bar with zeros, for example, just to have some data. Now, I populated my Zarr, my group bar with array buzz, with the zeros. In this case, what I can do, I can just call it like bar with key buzz. In this case, I got this information. And notice that all of this hierarchy, it is already drawn here. So from the root, I got the foo, I got bar, and finally I got buzz. And this, by just storing these key and values, I got all of these locations. And the interesting part that it is literally folders with such a names, with this .z array inside them and z metadata inside them. Foo is also available, bar is also available here. Yes, and basically by taking the info of this bar array, we can see that, okay, there is bus inside of this one array, to which we can have the access. And finally, the root also could be accessed by keys. You can just pass them one by one or just combine them. And in this case, once again, we got our array with zeros. The same one here, like image store with the 0 level and the first level of pyramid will produce us the array itself. So now we can have the access to it. We can simply call the method tree, and for root it looks like this: the root, foo, bar, and bus inside of it. And if I will get to the image store, let's look like this: 0 level and levels of the pyramid with the decaying size of the pyramid itself of each array. So in this case, whenever I will use this image for the visualization to produce it to the pathologist, they can simply use all of these levels just to rapidly zoom in and zoom out, and this data accessed very, very fast. Additionally, what I can do, there is in metadata attributes. For example, I can populate the attributes of foo with name bar, and it will be cached here. So now I can access it from here directly. And the interesting part, and I would say that this would be the final part, let's not discuss the AnnData itself, because we had only 10 minutes. It's the rendering time based on the chunk size. This would be the last image, but this is also important about Zarr, is that the chunk size and how we should optimize it, it highly depends on the time to render. Of course, the larger the chunk size, the harder you can compress the image itself. So if you want to keep a lot of the space on your disk, in this case, you can, of course, choose the large chunk size. But usually this is not impossible. And for the sake of simplicity and quick access, usually you can follow the size of chunks somewhere between from 4 to 16 megabytes of your data. And in this case, this is already the render time. So this is profiled render time of the zoom level 0 of the pyramid, 1 of pyramid, and etc. And you can see that here, for example, let's zoom in, at least if it's possible. Because on the level 0 it's observable pretty good. With this low size of the chunks, this time to render is actually crazy. It's like hundreds of the milliseconds for each chunks. And all of these chunks are loaded in parallel. It means that you will just on spot have all the data, and you will load all the necessary data to show to your pathologist or whomever you need to show it. And that's why basically NASA adopted it, because whenever you're going into these very deep images and they got crazy sizes of pyramids, you will render it just in a second. So yes, I guess that's it. Yes, I already marked this. We have not enough time. So that was basically it for the self-supervised learning for the Zarr format. Zarr once again got a huge number of the documentation. It's well documented. It got synchronizers. It got integration with Dask. So we can just go to documentation and adopt it to any format you want. It's also an open source format. It means that it's rapidly evolving, and usually not me directly, but but tech leader in our team submitted an application, opened an issue on GitHub to these Zarr developers, and they just closed it in maybe 2 weeks. I mean, successfully with the problem resolvance, which is also pretty good. And I mean, not every developers could say that they can do so. So thank you, everyone. That's it from my side.